Okay, primary and non-primary cells. Primary culture cells are immediate cultures of cells taken from an animal. Primary cells will go through a few rounds of cell division and then die. So these are cells taken directly from normal tissue and then moved to an in vitro environment like a plate or petri dish, etc., and then grown in that environment. They will go through a few rounds of division and then they will die because normal cells cannot indefinitely go through mitosis. Non-primary culture and cell lines, non-malignant cells. So these cells are taken from normal tissue, but we have the issue that we have with primary cells right here where they will die after a few rounds of cell division. So what you do is when you take these cells from normal tissue, you transform them. And this process of transformation is essentially making them cancer-like. The process of going from normal to malignant or cancer-like is called transformation. Okay, so what you're doing is you're taking these normal cells and you are going to transform them. And the way you do that is you introduce genes that cause cancer. So oncogenes are one way. Or you infect them with viruses that cause cancer. So cancer-causing viruses. And once they're cancer-like, they can divide as many times as you wish and not die. Non-primary cultures and cell lines of malignant cells. So in this case, what we're doing is we're taking cells from tissue that is already malignant. So these cells are not going to have any trouble dividing in culture. They will divide and divide and divide and not die because they're malignant. They're already cancer-like or they're cancerous. So those are the three types of cells. Typically in a lab, you don't work with primary cells. You'll work with the other two that are either normal cells that have been transformed or cells that have been taken from tumor. Now, in the case of number 2 and 3, even though the cells are transformed after being taken out of the host, or they were already transformed, meaning they were taken out of the tumor, those cells still have the characteristics of the tissue they originated from. Of course, their phenotype will change once they get into the in vitro environment because of dedifferentiation, but they still have the phenotypes or characteristic phenotypes of the tissue that they originated from. Established non-primary cells have a name, and a stock of low-passage cells can be purchased from vendors such as ATCC. So all cell lines have a certain name, and there are cell lines that people normally work with that are well known, depending on the origin of the cell line, etc. And they have names associated with them. So people will know, for example, if I say I'm using BHK cells, which were going to be the cell types you were going to use in lab this semester. They're baby hamster kidney cells. There are certain type of baby hamster kidney cells, and that cell line exists in the research community, and everybody knows that if you have that cell line originated from a single sample of kidney cells from a baby hamster. So it's not that every baby hamster kidney cell is the same. It's just saying that cell line originated from an original bunch of cells, an original culture, and then it was expanded. Now, what do we mean by what I just said? You take some cells and you expand until you have almost an infinite supply of cells. So if you start out with one culture of cells, and these cells, of course, have been transformed or were already transformed, and here's my plate of cells, right? And what you can do is, and we'll call this passage 0, okay, because we haven't passaged them yet. And what you can do at this point, once you get the cells and they grow, and the bottom of the plate is completely covered with cells, because these are going to be of epithelial origin, so you're at 100% confluency, or near it, 80-90%. Well, at that point, you have to split the cells. So, for example, if at this point I go ahead and I split this 1 to 5, now what I can do is, these are going to be passage 2, by the way, or passage 1, I'm sorry, passage 1 cells at this point, right? Let me move this over here. Okay. So what I can do at this point is that each one of these plates, right here, 4 of them, I can go ahead and freeze them down into 4 vials. So what I'll have is 4 vials of cells at passage 1. Each vial is representing one plate of 80 to 90% confluency cells, right? So it's plenty of cells. Now what I can do is then take these passage 1 cells and go ahead and split them 1 to 5 when they get to near 100% confluency, right? So 1. And I go ahead and I freeze down these 4. And these are going to be passage 2 now. Okay. And I go ahead and I have now 4 vials at passage 2, right? Now I can keep going like this. Now I'm being very conservative here. I'm only doing a 1 to 5 split. I can do a 1 to 10 split. It depends on the cell type, but I could go from 100% confluent cells and go to a 1 to 10 and actually freeze down 8 and then do another 1 to 10 with 2 other plates and keep going like that and have many more vials of cells. But essentially what will happen is that I will have 4 vials then, if I keep doing this, right, of passage 3, 4 vials at passage 4, and while I'm doing this, I'm trying to figure out for whatever I'm studying, whatever system I'm studying that I'm using these cells for, at what passage number does that system sort of go awry and I can't go past that passage number. Now, if that passage number is, for example, passage 30, well, I'm going to have all these vials down from, passage 4, etc., to maybe passage, if it's passage 30, then I'll say, okay, passage 20 is a good place to start if I'm for my experiments, right? So what I can do is once I get passage 4, at this point, I can basically maintain these cells kind of in the lab for a while if I have a passage 20 cells, because passage 30 is sort of my limit. I'm not going to use anything above passage 30 for my experiments. So I get to passage 20 and I can split them 1 to 5 and then again 1 to 5, and then I have all these cells that I can work with for my experiments in the lab and etc. And maybe I can, once I do that, when I do all these big splits, I can start to stockpile a bunch of vials of passage 21, 22, and 23. And if I run out of these passage 20 cells, I can always come back to here and unfreeze some, right? And then if I start feeling like I'm running out of 20s, I can go ahead and unfreeze some passage 4 or the passage before, passage 19 or passage 18 or etc., and keep expanding. So what happens is once you have that 1 vial of cells, you can basically make an infinite supply of them, of those cells. You have to find out what passage number is your limit and then work from there. Now, as I said, I'm being very conservative here because typically what I would do is I'll take 1 plate and do a 1 to 10 split. And some cell lines, they grow really fast, so you can actually do a 1 to 15 or something like that and immediately have a bunch of cells to freeze down. And really, instead of 4, 4, 4, I could have 10, 10, 10, and just have all these cells put away at different passages and always be able to work my way backwards. You get 1 vial of cells from somebody, you buy them, etc. You should be able to make an infinite supply for your lab. And that's basically what I did here when I got to UT Martin. I just got some donated cells from some of my friends, and I just did this whole process so that we have infinite supply of these cell lines at UT Martin. All right. And that's something that you can't do with primary cells, because primary cells, as soon as they go through a couple of rounds or 3 or 4 rounds of cell division, they die. So it's impossible to do that. That's why you need either cells that have been taken from normal tissue and have been immortalized, made cancerous, or you take cells that are from a tumor. All right, so genetic instability. At every round of cell division, there is a small probability that a mutation can occur. This probability is increased many folds for cells that originate from tumors. In a malignant transformed cells, many genes whose products are involved in preventing mutations are inactivated. And this is true because one of the few things that have to be inactivated in a cell if it's going to have the cancer phenotype is that you have to inactivate any DNA checkpoints, meaning the checkpoints that if there's something wrong with the genome, the cell will either stop dividing, try to fix it, or it will go through apoptosis, programmed cell death. So those are inactivated. Also, the normal process which will activate programmed cell death, apoptosis, has been inactivated through mutation. So we got a kind of a double whammy, right? Apoptosis is kind of inactivated, and the DNA repair process is inactivated. When normal cells go through cell division, and if their DNA has been compromised, their chromosomes have been compromised through major mutational events, these proteins that are involved in monitoring for mutations, they stop cell division right in the S phase or earlier, and they will give time to the cell to try to fix the problem. Now, if the cell is unable to fix the problem, the cell will be shoved into programmed cell death and apoptosis, because when you're looking at living tissue, there are millions and millions of cells. It's better to remove that one that can possibly become a cancerous cell than to let that one somehow stick around, because if a cell is gone through apoptosis, you can replace that cell easily by a normal cell going through mitosis. So for a cell to become cancerous, to go through the transformation process, one thing that it has to do is remove the checkpoints, the proteins that are involved in monitoring for mutations. And the second thing is to inactivate apoptosis, or as many triggers for apoptosis as possible. A normal constituent of cell culture media is fetal bovine serum or other types of serum. Serum contains growth factors which are consistently pushing the cell through the cell cycle. So in the normal solution that your cells or cells of any animal is bathed in that normal in vivo environment, has a lot of growth factors and also factors that simply tell the cell to stay alive. So if when you bring the cells from that in vivo environment to the in vitro environment, you have to provide those same factors, growth factors, what we call trophic factors, which is not written here, but trophic factors. Trophic factors, what they do is they're giving signals to the cell just to stay alive. In the absence of trophic factors, the cells will go through programmed cell death. So to get that source of serum, it would be very difficult to get that from humans, because you would need a lot of blood transfusion, etc., to get enough serum to be able to do your work. So the source for serum comes from, cows and calves, and this comes from the slaughterhouses. So the slaughterhouses will sell fetal bovine serum to companies and fetal calf serum to companies, and then those companies will process it and kind of clean it up a little bit, make sure there are no microbes or etc. in it, and then sell it to the researchers. So what you do is when you have your media, the regular media, which we'll talk about shortly, that is sort of this reddish liquid, that has all the carbohydrates, amino acids, and cofactors and vitamins and buffer and pH indicator and those good things in it that the cell definitely needs. And what you do is you add the serum to that media to complete it. So you get complete media. Complete media has serum in it. So that now the cells have all the goodies they need to survive, meaning the food and the monomers and the cofactors and etc., but they also have the signals coming into them to stay alive and also divide. Therefore, the genotype of cells in culture will change over time. A genotype change will lead to a change in the cell's phenotype. So when there is genetic instability, the place where mutations will occur when you have genetic instability, meaning that the checkpoints are gone, nobody's monitoring the genome anymore, and there's something going on in the cell that creates even more mutations to occur. The rate of mutation has increased. So where mutations occur is when DNA is being replicated. And where is DNA being replicated? DNA is being replicated during the S phase of mitosis. So every time the cell goes through cell division, it picks up more mutations when it's transformed, okay? So since the cells are constantly being pushed through mitosis because the fetal bovine serum or the fetal calf serum has growth factors in it, then at every round of division, you're getting more mutations. And that's why we monitor the passage number, because the passage number is sort of a rough estimate of how many divisions the cells have gone through. And you know after so many divisions, the genotype is going to change so much that will lead to phenotype changes. And those phenotype changes could be ones that basically render the cells ineffective for the experiment that you're doing. So high passage number cultures can behave very differently than the cultures of the same cells at lower passage number. It is important for researchers to test at which passage number a phenotypic change occurs that will affect his or her experiments. And we've talked about these examples here, so I'm just gonna let you read them on your own, but we talked about this virus example. It is important not to confuse genetic instability with dedifferentiation. There are two separate processes that must be considered when designing experiments. Dedifferentiation is literally the process where I take cells from an in vivo environment and I move it to an in vitro environment, and there's a phenotypic change because of the environmental change. Genetic instability is more of a long-term event, meaning that as the cell divides and goes through mitosis, it keeps picking up more mutations, and eventually you have enough mutations that you start getting a phenotypic change that affects the results of your experiment, and you have to be on the lookout for that. Okay guys, we'll stop here and continue later. Thank you.